Hi guys, welcome back. As you all know, I'm Mansa, currently pursuing my BE in Sri Venkateswara College of Engineering. So right now I'm in my fourth sem. So today I'm going to explain about the subject biology for the computer engineers, and the topic I have chosen to explain in this particular video is protein structure. So where we go to see the introduction to protein structure and the types of protein structures, where you get to see the levels of protein structures. So first let us get into the topic, that is introduction to protein structure. So proteins are essential macromolecules that perform a vast array of functions in biological systems. Understanding protein structure is crucial for deciphering their function and interactions. Proteins are composed of one or more long chains of amino acids which fold into specific three-dimensional shapes. Level of protein structure. So protein structure is categorized into four levels, like namely primary, secondary, tertiary, and quaternary. Each level of the structure is critical for the protein's overall function and stability. The sequence of amino acids determines the higher levels of protein structures through various interactions. So let us see the primary structure here. So the primary structure refers to the linear sequence of amino acids in a polypeptide chain. So as the name itself indicates, it's the linear structure or the single structure that consists of it. So it is called as primary structure. So this sequence is determined by the genetic code and dictates all subsequent folding and interactions. Any changes in the primary structures can significantly affect the protein's function and activity. So there should be no changes in the primary structure that takes place. In such cases, if that happens, it would affect the entire protein's function. So let us see the secondary structure. So here the secondary structure involves local folding patterns stabilized by hydrogen bonds between backbone atoms. So basically it can be seen in the backbone atoms. Common secondary structures include alpha helicals and beta sheets, which form a distinct shapes. So in secondary structure, we got to see two types of structures, namely alpha helicals and beta sheets. So these structures contribute to the overall stability and functionality of the protein. So tertiary structure. So tertiary structure refers to the overall three-dimensional shape of a single polypeptide chain. So as the name itself indicates, it's an overall three-dimensional structure, that is a 3D shape of a single polypeptide chain. So this structure is stabilized by various interactions including hydrogen bonds, ionic bonds, and hydrophobic interactions. So basically this tertiary structure is stabilized by types of bonds or types of some actions or the interactions, like namely we could see hydrogen bonds, ionic bonds, and hydrophobic interactions. There are many such things like that, like van der Waals forces and all, it is not mentioned here, but it is also helpful. The specific tertiary structure is crucial for the protein's biological activity and interactions with other molecules. Quaternary structure. So, quaternary structure refers to the assembly of multiple polypeptide chains into a functional protein complex. So, in this quaternary structure, it refers to the assembly of multiple polypeptide chains. So not all proteins have a quaternary structure, has some function as monomers. So most of the times in some cases there is a less chances of having a quaternary structure, because most of the proteins will be considering monomers, where we get to see only the primary structure of the proteins. So this level of structure allows for a cooperative interactions between subunits, enhancing functionality. So basically this structure will enhances the functionality between the subunits. So protein folding. What does protein folding means? So protein folding is a complex process that enables proteins to achieve their functional conformation. Misfolding can lead to loss of function or disease such as Alzheimer and cystic fibrosis. So Siafron proteins assist in proper folding by preventing aggregation and promoting correct conformation. So basically even the protein folding also matters when it comes to the protein structure and protein's functionality. If there's a misfolding, then it would cause many diseases that would affect the human body. Techniques for studying protein structure. Various techniques are employed to determine protein structure, including X-ray, crystallography, and NMR spectroscopy. Cryo-electron microscopy has emerged as a powerful tool for visualizing large protein complexes. Understanding the structure, function, relationship is enhanced by these techniques, providing insights into biological processes. So various type of techniques are involved in determining the protein structures, like as we see it in our daily basis and all, like x-rays, or you can even see in a crystallography and even the NMR spectroscopy and all. So basically there are more number of techniques where you can get to see about the protein structure, and where you can come to know about the protein structure and all. So importance of protein structure in drug design. So in drug design, what role does a protein structure performs is knowledge of protein structure is vital for rational drug design and development. So targeting specific protein structures can lead to the creation of more effective therapeutics. Structural information aids in identifying potential binding sites for drug molecules, enhancing specifically. So basically here we get to know about what the protein structure impacts in the drug design. So knowledge of these protein structure is can play a vital role in the drug design. So targeting only the specific protein, like if you target a one particular protein which is very useful, it would help or benefit the therapeutics and all. So let us see the conclusion of this complete protein structure. So protein structure is fundamental to understanding biological mechanisms and disease pathology. So it is basically used for understanding biological mechanisms and also disease pathology. So what causes, or what loss or what misfoldings would cause to which disease. So advances in structural biology continue to reveal the complexities of proteins, interactions and functions. Continued research in this field has profound implications for medicine, biotechnology and synthetic biology. So by this I'll end our video. Thank you for watching.